Okay, well, today I'm pleased to present to have Senta Georgia come from to give us a talk from at USC, University of Southern California, where she has been an assistant professor. Well, it was since the mid 2013 or so. Basically, she started off her career or spent her entire career studying beta cells in the pancreas, and where she started that in 2007 when she got her PhD, and then she moved over to the UCLA and continued on with a postdoc there, and then moved down to USC, where she's been ever since. And she's going to go and tell us about this really cool story that I heard cursory bits about from Mariah Sweetwine in our lab or in our department about COVID and diabetes. So I thought that would be a really cool thing and timely to hear about. So please take it away.
Thank you so much for that introduction. It is a pleasure to be with you all today. I wish I could be in person, but who knows when it's safe to travel and not, and all of this kind of stuff, and masks, et cetera, et cetera. So I'm disappointed I can't be there to see you all in person, but I really appreciate you taking out the time to come and listen about my work. And I look forward to getting any feedback or suggestions or questions that you have. Please feel free to stop me. I'm not going to be able to see the chat, so just interrupt if you want. Okay? All right. So just to outline my talk really briefly, we're going to talk about an existential question in this presentation, and I thought that was obvious what that would be. And then why is this actually a relevant question to us all? And is there any precedent for worrying about this question? Then I'll give you my answer to this question, and we'll talk about if my answer has implications for public health. Okay, so like I said, we're going to talk about this existential question. And it's the one thing I want to know is what comes first, the chicken or the egg? And when I asked the internet this question, it actually gave me some very obscene answers to which I was not prepared for. So that's not professionally appropriate. So I'll give you some of the non-profane answers that they gave. And this is me trying to figure that out. And then someone did it. Yeah. Okay. So the question is, which came first, the chicken or the egg? And the answers could be the egg because breakfast comes before lunch, and then clearly eggs because feathers were in the evolution of dinosaurs, and then there are some other ones. And if you just look it up in the dictionary, chicken comes first, right? But my favorite, because this is how I live my life, is that the Amazon will let me know, okay? Because I ordered one and it will eventually come in. But in all seriousness, in the beginning of the COVID-19 epidemic, there was a lot of speculation in the field that COVID-19 might actually cause diabetes. And so it was a big splash in the New England Journal of Medicine, and then in some of our clinical journals. There were cases of patients coming to the emergency room and diabetic ketoacidosis that was precipitated by COVID-19. It was in Science, it was in Nature, and I think it was Daniel Drucker set up the possibility of this bi-directional relationship between COVID-19 and endocrine dysfunction, specifically in the insulin-producing beta cells. So this was a lot of speculation in the beginning of the pandemic. But why was it of importance, I think, is the question. And it's because 1 in 10 people in our country has diabetes. And so that's about 34 million people in the United States. But what's crazy to me is that 22% of that 34 million people, about 7.3 million people, do not even know that they have diabetes. So that's 1 out of every people in the United States, and actually 1 out of every 4 adults over 65. And so we've seen this trend over time that I'm sure you've seen again and again, where this map is getting more and more dark blue, and that's the dark blue represents higher and higher concentrations of people who have diabetes. Okay, so it's concerning because if 1 in 10 of us has diabetes now, and at this point, 60% of us have been infected with COVID-19, and COVID-19 causes diabetes, that could severely impact the number of people who have diabetes in the future, thus putting a huge strain on our healthcare system and posing a unique threat to our national security. This is also concerning because there are people who have type 1 diabetes, separate from the number of people who have type 2 diabetes. And about 200,000 children and adolescents are diagnosed with having type 1 diabetes. But even, so we have the bulk of people who have type 2 diabetes, a subset of people who have type 1 diabetes, but then there are 88 million people in the United States that have prediabetes, meaning that they have a fasting hemoglobin A1C between 5.7 and 6.4. And this is especially prevalent among racial groups, but diabetes tends to be more prevalent in Black and Latino populations. So even though prediabetes seems to be spread out evenly, minority populations are disproportionately affected with having diabetes. Those same populations are also disproportionately affected with having COVID-19. And so this is a problem. And the reason that I think about it in terms, I mentioned national security, is because of the way that we've seen that we have to protect the infrastructure of our healthcare and our capacity to take care of people in the emergency setting during this pandemic. But we also have to start to think about how can we care for more people that are going to need more intensive levels of treatment as our population grows, and unfortunately may be growing in a direction where people are more sick. Okay, so there's this question as if, or there's this question, does COVID-19 cause diabetes? And we don't necessarily know. But there was some evidence from the first SARS-CoV, it wasn't a pandemic, but from the first SARS-CoV, why am I forgetting this word right now? But back in 2003, when the first version of a coronavirus was found, and it did not become a pandemic, but it was prevalent in China. And so what a group did is they matched 39 SARS patients who had no previous diabetes and received no steroid treatments while they were being treated with SARS, and compared them to their 39 aged matched healthy siblings during a 3-year follow-up. And 20 of those 39 patients were diabetic during hospitalization, as characterized by their fasting blood sugar levels. And so here is just a graph of their fasting blood sugar levels. And so for the control patients and patients that did not have SARS, they had normal fasting blood sugar levels below 5.7. But for patients that were infected, for at admission and 2 weeks after discharge, or 2 weeks after hospitalization, they continued to have elevated blood sugars. And then after discharge, the bulk of patients returned to being normal glycemic, but 6 patients still had diabetes. And after 3 years of follow-up, 2 patients still were diabetic. And so that's about 10% of these patients ended up having diabetes after 3 years. So now moving forward to the clinical picture, during this pandemic, there's been lots and lots of clinical reports that show that people who already have pre-existing diabetes have worse outcomes from COVID-19. But as the pandemic progresses, we're getting some nuance to that. And so just because you have diabetes is not necessarily a poor predictor of prognosis. If you have diabetes and your A1C is under control, you're generally okay. It's hyperglycemia at admission that is associated with the severity of the prognosis for patients with COVID-19. And so being hyperglycemic is a negative predictor of outcomes for people who have COVID-19. Now, this idea that what is hyperglycemia telling us? It can come from multiple things, right? So you could have pre-existing diabetes. You could just have stress hyperglycemia. You're sick, you don't feel well, your blood sugar is up. That's completely normal. It's also possible that the steroids that are given as part of the course of treatment for COVID-19 drive steroid-induced hyperglycemia. Steroids are toxic to beta cells. You get beta cell injury, you have some beta cell loss, but it's mostly a loss of function. And then post-steroid use, depending on how intensive that steroid treatment is, most of that function can be recovered. But it's also possible that we have beta cell dysfunction, and that beta cell dysfunction may be driving this hyperglycemia, but it's not clear, okay? In addition to just focusing on hyperglycemia and diabetes as an acute symptom of COVID-19, our attention is starting to shift to what they call PASS, the post-acute sequelae of COVID-19. And so at the time, I think this statistic is from February, there were 33 million survivors of COVID-19 in the United States. And about 25% of those survivors of COVID-19 have post-acute sequelae. And a recent report indicated that there is a 1.41 odds ratio of the patients that have PASS having new onset type 2 diabetes after a mild to moderate case of COVID-19. Mild meaning that you didn't even go to the hospital at all, and moderate meaning you may have gone to the hospital, but you didn't get anything more than light steroid treatments or antivirals, okay? And then another report that was just released at the end of last year also suggested that 10 to 20% of pediatric patients that have COVID-19 end up having post-acute sequelae of COVID-19. And so these numbers are starting to be troubling as we're trying to understand if this hyperglycemia during the pathogenesis of COVID-19 is inducing some sort of injury that can't be resolved that maintains this hyperglycemic phenotype. At Children's Hospital, we have done our own study looking at our patient population. And so what happened was, in the beginning, I would say sometime towards the end of 2020, my colleagues at Children's Hospital in our Department of Endocrinology, while we were having our faculty meeting, kept saying, I've never seen so many kids come in DKA. This is just crazy. These new onset kids are coming in DKA, new onset type 2 kids. And so we did a retrospective chart examination to try to understand if this was actually a new phenomenon, a phenomenon that was actually happening that was statistically significant. And what we found is that we looked at 2018, 19, and 20 in the months between March and August. And we chose those months because that was the data that we had thus far. And the appearance of diabetes can be cyclical. So we wanted to compare the same months over multiple years. And so what we found is that a total of 4 kids presented in DKA with new onset type 2 diabetes in 2018, and 2 presented in 2019. But by 2020, we had 16 kids who had come in, which was about 20% of our new onset type 2 diabetes for kids that had come in. And of those kids, some of them were severe DKA. None of them were, only one was positive for a pancreatic antibody, but the majority of them were not positive for pancreatic antibodies. And because this was early in the pandemic, We did not run SARS-CoV-2 PCRs or IgGs on every patient that came into the hospital. But of the patients that we did see in clinic, 14 of those patients in DKA ended up getting a PCR test, and they were all negative, so they were not acutely infected with COVID-19. But of the 6 that had the IgG antibody test, 2 out of 6 of those patients were positive for the antibody test. And so that was also very concerning to us in this report, because this is before vaccinations, and none of these kids had severe COVID-19 to the point where they actually knew that they were had COVID-19. They were not hospitalized. So this started to lead or contribute to the idea that new diabetes may actually be linked to COVID-19. So this was a story in the Washington Post. This was a story of even posted by Dr. Francis Collins, the director of the NIH, thinking about how COVID-19 can lead to beta cell dysfunction and lead to diabetes. Okay. And now there are more and more studies coming up that are actually giving guidelines to suggest that we need to study this more intently. And it isn't just type 2 diabetes, but especially for children, it might also be type 1. And so the clinical data is telling us this, but the actual field of beta cell biology is in a lot of debate. And the best type of study to do is when there's conflict in the literature. And so in early 2020, Shubing Sheng's group from Cornell published a paper where they differentiated human pluripotent stem cells into multiple cellular cell lineages, but specifically into beta cells is what we'll talk about. And they used cadaverically acquired human islets and subjected them to in vitro infection with SARS-CoV-2, and then did all sorts of analysis and found that both beta cells derived from stem cells and cadaveric islets are permissive to SARS-CoV-2 infection. Okay. Then a paper came out from a group in Italy that suggested that human beta cells do express the receptor for SARS-CoV-2, ACE2. But then 2 groups, one out of Vanderbilt and the other one out of the University of Florida, took sections from pancreatic histology sections or pathology sections from patients who had expired from COVID-19, 3 and 4 patients respectively, and had done analysis and decided that there was not significant ACE2 or TMPRSS2 co-expression in islet endocrine cells, and did not think that SARS-CoV-2 was directly infecting those beta cells. And so, like I said, that was out of Vanderbilt and out of Florida. And so these 2 groups thought that the direct infection of pancreatic endocrine cells via ACE2 was unlikely to be a central pathogenic feature of COVID-19 as it relates to diabetes. Then a group out of Germany came up and had taken more pathology sections from patients that had expired from COVID-19 and showed that you can actually see the virus in the cells of the exocrine and in the endocrine pancreas ex vivo and in vitro. So their only in vivo data was using these histology sections and detecting the viral replicative capsid in the tissue. Otherwise, what they did was to use, again, human isolated islets and stain for multiple markers and show that there looked like that there were morphological changes in the beta cells, transcriptional changes in the beta cells, and a reduction in functional secretion. Okay, and so the overall takeaway from those studies were that SARS-CoV specifically infects beta cells in vivo and in vitro, reduces glucose-secreted insulin secretion in vitro, causes beta cell death in vitro, and shows that the viral protein induces apoptotic kinases. So that is what this showed. So when we started our study, we wanted to be able to resolve this conflict in the literature. And so we wanted to pose the question, does SARS-CoV-2 infection cause beta cell dysfunction? And we hypothesized that yes, it did. But human samples are hard to get and hard to analyze from patients who have expired of COVID-19. One, any pancreas that you get from an autopsy study is generally one of the last piece organs that are collected. And it unfortunately is one of the first organs to undergo autolysis because of the nature of the exocrine pancreas. So your tissue sections are generally just not very good. Second of all, the only people that you're getting SARS-CoV-2 or that you're getting pancreatic sections from will be people who died from COVID-19. And the thing is that 90% of people are mild to asymptomatic with that infection. And as such, people who have died of COVID-19 have had to undergo multiple treatments, heavy doses of steroids, and are basically represent a very confounded population to study. And so we wanted to approach this from a position of having a relatively mild case of COVID-19 and trying to understand the pathogenesis of COVID-19 in relationship to beta cells in a mild model of COVID-19. And so we partnered with a company called BioQual, and BioQual is a research services company. We were introduced to BioQual through the COVID Consortium, which is a group of laboratories doing viral detection in macaque studies and doing field testing for different COVID-19 tests at the time. And so BioQual was doing a lot of the preclinical testing for vaccines and other small molecules to treat COVID-19. And so they had recently published in Science their model of in vivo COVID-19 infection or SARS-CoV-2 infection. So what they did was they would inoculate rhesus macaques intranasally and intratracheally with SARS-CoV-19. We call that day 0. They could detect the highest viral load in their bronchial alveolar fluid by day 2. And then they would euthanize their monkeys at day 7, 10, or 14. But by day 12, these animals had had the resolution of their clinical symptoms. But overall, their clinical symptoms were relatively mild. They did not require any sort of intervention. You could tell for the first 3 to 4 days, the animals were sluggish, but as in they didn't feel well, but otherwise didn't require any acute care. And by day 12, most of the resolution of clinical symptoms had occurred. So they would take these animals and euthanize them along the way of this 14-day protocol for their own purposes, which would totally ignore the pancreas. And so they would collect the pancreas, throw it in formaldehyde, because formaldehyde inactivates SARS-CoV-2 and makes it possible to process tissue at a BSL-2+ facility, and then send it to us. So the first thing we wanted to do as we embarked on this study, trying to understand was SARS-CoV-2 able to infect beta cells in vivo, using not some model where you take the islets out and just bombard them with a virus and just try as hard as you can to get it in there, but in the same sort of way that patients get SARS-CoV-2 through an intranasal or intratracheal infection. And so the first thing we needed to do was to see if ACE2 was expressed in the beta cells of primates. And so what we did was do a single cell sequencing analysis on a pre-existing data set from rhesus macaques. And what we did was to isolate the cells that were pancreatic, and then did some clustering to understand which of these cells express TMPRSS2 and ACE2. And then we broke that down by the subset of cells that are in the pancreas, and what you can see is that beta cells are here in red, and most beta cells express TMPRSS2, and a subset of beta cells in the macaque also express ACE2. So then we moved on to histological analysis, and we obtained our control animals not from bioqual because all of their animals were infected. And so their control, because their experiments were trying to understand if their interventions, be it a vaccine or a small molecule, were effective, their controls were the animals that were infected and untreated. So we had to source animals from the University of Oregon's Primate Center for our controls and some collaborators that we had at the University of Wisconsin in Madison. And so here we can see if we stain the pancreatic islet, denoted here by insulin staining in green, with a receptor for ACE2, we can see that there is ACE2 staining in beta cells of the pancreas, as we can see with this overlap. So once we showed the ACE2 expression was present in beta cells, the next question we wanted to ask was, is SARS-CoV-2 present in the beta cells of the acute and the post-acutely infected primates? And so here in red and blue is DAPI and red is an antibody against the spike protein. And in the acute and in the post-acute, you can see that there's a red kind of spread out throughout the islet, but in the control animals, we don't see that. And so when we overlay that with insulin, you can see that insulin is overlaying with these pockets of red staining, suggesting that these insulin positive cells are positive for that spike protein. So we can see the spike protein by immunohistochemistry, and that suggests that the virus is present, but we can see viruses using electron microscopy. So the next thing that we did was to assess if we could, or to actually detect evidence of viral capsids in the beta cells. And so this is a sample from an acutely infected animal. This is within an islet. We have multiple cell types here, but this cell has ruptured. And you can see that this is called a viral replication complex. And so you have this sea of kind of empty vacuous space here. And we don't think that this is an artifact of tissue processing because we don't see these vacuous spaces in these surrounding cell types. And then when you zoom in to this viral replication complex, you can see multiple viral particles that are being developed or formed, packaged in this viral replication complex. We also see the presence of these double membrane small vacuoles that are also indicative of viral replication in beta cells. We went to go back to the literature to see if that's what it looked like in these cells that people had, in the islets that people had infected with SARS-CoV-2. And so this is just an example from Mueller's paper that came out last year, the German group, where you can see they also have these large vacuoles that are kind of like replication complexes with the virus particles in between. So we know at this point that SARS-CoV-2 is present in the beta cells of the primates that were infected with SARS-CoV-2. So then the question is, how does that affect beta cells? And the first thing that I would think of as a beta cell biologist is to measure beta cell mass, right? So what we did is to use our histology sections and stain them just for insulin and for DAPI. And we used DAPI to get a sense of the tissue morphology and then take bidex images of our tissue section and overlay the DAPI on top of our insulin staining so that we can get a sense of, and not just get a sense, but quantify the percentage of the tissue that is taken up by insulin area. And what you can see is that in our control tissues, the beta cell area is about twice that of the acute and post-acute samples that we found. And And so beta cell area can be affected by either, or beta cell area can be decreased by either beta cell loss, which we can measure usually by apoptosis, or beta cell atrophy, which is that shrinking of individual beta cells. And so the next experiment we pursued to try to understand that is to actually measure the size of beta cells and stain for apoptotic markers. And what I can tell you is that I didn't show you the pictures, but when we stain for apoptotic markers, we don't see any difference in the expression of cleaved caspase 3 or in tunnel expression in these samples. So next what we did was to measure the actual size of the beta cells. And so we stained for insulin, and then we also stained for a marker that outlines cell outlines, like E-cadherin, and so that we could detect the cell membranes. And we draw a line around that, and we calculate the area in between that line. And so when we do that, we can show that there's actually a statistically significant stepwise decrease in the size of beta cells throughout our study period. So the control beta cells individually are much larger than the acute and post-acute beta cells. Another way to understand the relationship between beta cell loss versus atrophy is to look at beta cell or islet cellularity. Islet cellularity is what is the relationship, the ratio between beta and alpha cells. And so, or beta cells and the number of cells present, endocrine cells present in the whole islet. Beta cells in mice tend to be about 80 to 85% of all of the cells in an islet, such that if you see, even if you can't detect beta cell apoptosis, if you see a skewing of that distribution of the total percentage of insulin cells, then you know that you've lost the beta cells and you just missed that apoptotic event. And so what we did was count the number of insulin cells that are present per islet, and what we found is that the number of insulin cells did not change. So if the number of insulin cells didn't change, we don't see any apoptosis, but we do see this individual cell atrophy. We concluded that our decrease in overall beta cell mass is related to this cellular atrophy and not to the actual loss of beta cells. But what we did notice while we were doing all of this is that the beta cells from the control animals weren't just bigger, but they looked plump, like they were brighter. And so because we were working with fixed tissue, we couldn't just grind up the tissue and do an insulin content analysis to find out if the acute and post-acute pancreas had less insulin than our controls. So we actually had to do something new. And I work at Children's Hospital, which is part of USC. And I like to say that we have more microscopes than we do people most of the time. And if it's new and on the market, we're going to buy it. And so we have this amazing super resolution microscope. And what my graduate student figured out is that if she took high resolution images of the islets, you can physically see that in the controls, these islets are nice and full of insulin granules. But in the acute, we have these large spaces that are kind of just missing and black, similar to that large space that I showed you in that electron microscopy image. And so what she did was to measure the number of maxima. So what that meant by meaning maxima is the number of green pixels in the area of the whole islet, and then divided that by the average size of the beta cell. And so then you get a measurement that is the number of pixels representing insulin, or number of maxima representing insulin granules per beta cell. And when we do that calculation, we can show that these beta cells from our acute and post-acute animals are severely degranulated in comparison to the wild type control. And beta cell degranulation is indicative of beta cell stress. In addition to their beta cells, or beta cell stress can be activated by multiple mechanisms. And at this point, we don't necessarily know what is being activated. I mean, we know it's being activated. We hypothesize it's being activated by SARS-CoV-2 infection, but we don't know what SARS-CoV-2 is doing. And so in one of the papers that had come out, they suggested that they saw markers of a mechanism of cell death that is different from apoptosis called necroptosis. And necroptosis is vastly different from apoptosis insofar as that apoptosis tends to be an acute, quick onset way to get rid of cells. Necroptosis can be a slow trod to cell death, and then they upregulate completely different proteins. And so because of that data, we wanted to look and see if we could see an increase in necroptosis markers and to see if maybe necroptosis was contributing to the stress, making the cells become degranulated and pushing them towards our loss of beta cell mass. So we stained for a marker called HMGB2A, and it's not the best stain, but what we took from this data is while it's expressed in most cells of the control, it is concentrated in the nuclei of these beta cells and of these cells that look like they used to express insulin and they don't anymore that are part of this islet. And you can see it concentrated in specific foci of the post-acute pancreas in a way that we don't see it expressed in the control pancreas. So it's possible that necroptosis may be contributing to beta cell loss, but it might be a slow process to actually get to that beta cell loss that we're not seeing within this first acute 2 weeks post-infection. So I just want to take a second to show you something that's not a piece of data that we have, but a future direction. Sorry if this feels a little bit out of place, but I want to make sure you understand why we think that this necroptosis may be important. And so one of the things that we want to do, and we just received some funding from the FDA to pursue this experiment, is to try to measure if we can find beta cell death in the serum of these macaques and of COVID-19 patients. And the way that we're doing that is in a collaboration with Raghu Miramira out of the University of Chicago, using his technology to measure the amount of methylated versus unmethylated cell-free DNA in the plasma. And so there is a CG in the insulin promoter, or in the promoter of insulin in humans that's basically always methylated in endocrine cells and not methylated in the rest of the tissues. I'm sorry, it's always unmethylated in endocrine cells and methylated in the rest of the tissues of the body. And so when cells die and macrophages clear those cells out, a little bit of DNA is released into the bloodstream as cell-free DNA. You can actually capture that DNA and then measure using digital droplet PCR the proportion of methylated to unmethylated insulin DNA that is in that cell-free DNA. And so what we're doing, or what we plan on doing with Dr. Miramira, is to look at the serum from our macaques and from our COVID-19 patients to see if we can detect unmethylated insulin promoter cell-free DNA in the plasma of these patients as evidence of them either undergoing beta cell death or not. And so that is data that will be forthcoming. And so I've shown you the beta cells in our model look degranulated, suggesting that they have beta cell stress and that we've lost beta cell mass, and that we have the expression of the proper receptor, and that the virus seems to be in the beta cells. And so all of those things are hallmarks, histological hallmarks of diabetes. In non-diabetic versus patients that have type 2 diabetes, there's a loss in beta cell size, there's a loss in beta cell number. We generally see evidence of degranulation or dedifferentiation in this transition between, in the differences between individuals that are normal glycemic versus patients that have type 2 diabetes, which made us start to think, like, when beta cells are degranulated, they're generally not functioning well. And while we can't measure insulin secretion at all from the data that we have or from our fixed samples, we can hypothesize, or we can guess that they most likely have a reduced insulin secretion, that they might be hyperglycemic from this, and then that hyperglycemia can't be addressed because there is impaired secretion of insulin. And in addition to there just being less insulin per cell, cellular metabolism is also intimately tied to insulin secretion. And there's a paper that came out 2 years ago at the beginning of the pandemic that showed that when macrophages and when some of the when cells are infected with SARS-CoV-2, what happens is that SARS-CoV-2 actually shifts their metabolism from an oxidative metabolism to a more glycolytic metabolism to produce metabolites that are necessary for viral replication. And so what we wanted to understand is, do we see any signs of either of something that would indicate that beta cell metabolism may be impaired and thus may be driving insulin reductions in insulin secretion? Okay, so here's another electron microscopy image from an islet from our acutely infected animals. And so here's a beta cell that doesn't show any signs of infection, these two here. But this beta cell here, we see these large lakes of empty space between the insulin granules, which we think is reflective of what we showed you in the previous electron microscopy image. But additionally, and I think what's important, is that the mitochondria are enlarged and blown out in their cores. And this is something that we see consistently in our acute and in our post-acute animals, as opposed to the cells that aren't looking like they're in distress and could be right next to those cells. The mitochondria integrity is intact and the size is normal. And so if your mitochondria are being lost and your cells are stressed out, they're going to use more glycolytic metabolism than they would use an oxidative metabolism. And we suspect that that is what might be driving this shift in metabolism. The issue for us is that in order for us to receive this tissue and be able to work with it, we have to actually fix the tissue. And I didn't want to do this in isolated islets. And so we can't use a technique like seahorse to actually measure metabolism. And so in partnership with Scott Fraser at USC and Jason Jung, what we started to apply the technique called fluorescence lifetime imaging to our histology section to try to understand the metabolism of these cells. And so how do we do that? So just as a really, really brief recap of metabolism, glycolysis happens in the cytosol. And the result of glycolysis is pyruvate, but it's also free NADH. That pyruvate then is translocated into, or shuttled, I should say, into the mitochondria to undergo the TCA cycle, where there is NADH that is produced, but that NADH is then bound by the enzymes that drive oxidative phosphorylation. Okay, so in the cytosol, you'll have free NADH, but in the mitochondria, the NADH that is the product of normal oxidative phosphorylation are bound to enzymes. And what fluorescence lifetime imaging can do is measure the difference in the fluorescence emission of NADH when you stimulate NADH at a particular wavelength, such that bound NADH has a much longer lifetime than free NADH and the decay of that natural fluorescence when you stimulate it. And so those lifetimes are dependent on whether NADH is free if it's in glycolysis, or bound if it's in oxidative phosphorylation. This part of the technology, not new. Doing it on sections, no one's done this. Okay, one person has done it that shows that it could be done in like a cell line, but not in fixed tissue. So how do we translate this this type of data into something that we can use? Like, how do we process that data? And so the way that this works is that, so I told you, we measure these lifetimes, and every pixel in a given image gets an intensity of the lifetime measurement, and is plotted on what we call a universal circle. And we can draw a line through the centroid of the mass of plots, or the mass of these pixels that we measure the intensity. And at the one end, at the upper left end, I should say, this T2 point, that is where, if NADH was 100% bound to enzyme, where it would land on the universal circle. And if it was 100% unbound to enzyme, it would land here at T1. So if you draw a straight line between those 3 points, then the position of this phasor on this line will give you an approximate ratio of bound versus free NADH. And this bound versus free is now indicative of the metabolism of a cell or of an islet, such that if your phasor plot ends up being higher and closer to the left, it's bound NADH, suggesting that your cells are more oxidative. And if it's lower to the right, that suggests that the cells are more glycolytic. So we did some control experiments with using cell lines just to validate this. And what we found is that cells that are growing in control conditions, these are just INS1E cells, have a higher glycolytic place, or what we call a glycolytic coefficient, than ones that are being grown in glucolipotoxic conditions. Okay, so what we did was we took our slides and we stained them with insulin. Then we took that image of insulin to create a mask, and then we measured the intensity of NADH, and we basically gated the intensity image with the insulin mask, so that we only measure the intensity, the fluorescence intensities, those lifetimes that were coming from insulin cells only. And so in these images, red in the first column is just insulin, but red in the second column means that there are longer lifetimes, and blue means that there are lower lifetimes, such that we can look at how these lifetimes fall out on a phasor plot and then calculate the glycolytic coefficient. And what we found is that our control animals grouped very separately from our acute and post-acute animals. We also, in addition to the animals that we had gotten from the University of Oregon's National Primate Center, we wanted to make sure that this was not just a artifact of viral infection. And so we obtained some samples of animals that had been infected with Zika and were pregnant. So pregnancy is inherently a condition of insulin resistance and requires more work from your beta cells. And in addition, these animals are infected with Zika. So if they had a reason for their beta cells to be stressed out, they would be. And so we could see that these control samples cluster very differently from these post-acute samples. Then we actually calculated the percentage of free versus bound NADH and could show that the control animals had less free NADH than the acute and the post-acute animals, suggesting that they had a more oxidative metabolism than the acute and post-acute animals. So That then asks the question, are these animals dysmetabolic? Are they secreting insulin properly? And I'm going to show you the raw data, and you tell me what you think. So here is a listing of the animals that we had blood samples from, and what their serum fasting glucose is, and what their serum fasting insulin is. And fun fact, insulin in non-human primates when they're fasting is 10 times higher than it is in humans. So the first thing that jumps out to me in looking at all of these is that this animal here just had diabetes to start with, right? But the rest of these animals are well within a normal glycemic range when I'm thinking about humans, where normal glycemic is basically anything under 99. Diabetes is over 126. And pre-diabetic, when you're fasting overnight, is somewhere in between 126 and 99. So all of these look relatively normal until you know what normal fasting glucose is for primates. And so doing some literature searching in a lot of the veterinary journals, I learned that fasting glucose levels in primates are about 20 to 30 milligrams per deciliter lower than they are in humans, such that when you go over 100, is most likely to indicate overt diabetes, and over 79 is more indicative of prediabetes. And so with that in mind, when we start to look at what some of these higher blood fasting serum levels are, you can see that there are some of them that actually have relatively high insulin levels in comparison to the other ones. And so it's not clear yet that in this acute phase that their blood sugar is exceptionally high and that their insulin is exceptionally high, but there does seem to be what has been characterized as dysmetabolic dysmetabolism for monkeys, where dysmetabolic begins at 60 milligrams per deciliter and goes up to 100, and diabetic starts at 100, and the corresponding insulin secretion values that go along with that. So taken together, we think that this project shows that SARS-CoV-2 infects beta cells, has consequences that seem to damage beta cells, specifically focusing in on a loss of mitochondria, and then that loss of mitochondrial capacity is reflected in a shift towards a more glycolytic metabolism, which then could suppress insulin secretion. And so we think that this should contribute to our understanding of the full spectrum of SARS-CoV-2 infections and help identify long-term consequences of the pandemic on the future health of affected individuals. And so going back to our first question, which is COVID-19 and diabetes, is it a chicken or an egg? Which comes first? Is it that you have diabetes, or maybe you have pre-diabetes and you don't know, and you get COVID-19 and it drives you into having a hyperglycemia? Or is it that you get COVID-19 and you have beta cell dysfunction and beta cell injury, and that drives you into being hyperglycemic? We're not sure yet, but I think that our data suggests that there is beta cell injury that could lead to beta cell dysfunction and compromise beta cell function such that it could drive hyperglycemia. So as for the question about the chicken and the egg, I don't know who comes first, and it doesn't really matter. And then I don't know if you guys remember these commercials about the most interesting man in the world. It used to be my favorite commercials on television, but it was also one of my favorite commercials on television because there is a very, very accomplished beta cell biologist named Alberto Hayek who really looks like that guy. And so if you are a beta cell biologist or know Alberto Hayek, you know Alberto has the accent. And don't you think he has the look? He has the look. But you have to know Alberto to care, I guess. So what are our future directions? We want to quantify the functional and metabolic effects of SARS-CoV-2 on beta cell function and survival in rhesus macaques over the long term. So not just in this acute 2-week post-acute phase, but in 3 months and 6 months after SARS-CoV-2 infection. And we want to determine if function and metabolism is restored. And we also have samples that were vaccinated against SARS-CoV-2 and see if that vaccination protects against the dysfunction in rhesus macaques. And we also are trying to develop a model to predict if diabetes is a post-acute sequelae of COVID-19 due to this beta cell injury. And so I'm just going to take one additional minute before we wrap up here to talk about this model that we're trying to develop. And so we were recently funded by the FDA to develop a model that integrates inflammation and tissue injury to predict and mitigate the post-acute sequelae of COVID-19. And so this contract is not just for looking at diabetes, but we're actually using the serum from patients, both pediatric and adults, to do clinical cell-free methylome profiling and couple that in the same samples with clinical proteomic discovery and with in-depth phenotyping of the patient to try to develop this model to predict which patients are more susceptible to having a post-acute sequelae of COVID-19. And then we have some in vitro and in vivo models to assess what's happening in tissue damage. And so our goals of that project are to produce a clinically predictive model that forecasts these post-acute sequelae based on disease-specific proteomic and methylomic structures. Signatures, not structures, and then hopefully by using that signature, we can deliver a list of potential druggable targets to mitigate that tissue injury during disease course. And so our cohorts are adults, pediatric patients, our primates, and some in vitro modeling where we integrate all of this multi-omic data as well as looking at our model data to do FLIM and image mass cytometry, validate these targets, and predict this model. So I would just like to say that my lab is amazing. The newly minted Dr. Kate Millett did the majority of the histology work. She's now left the lab and is a real working grown-up and my first PhD student, so I'm really proud of her. My newest PhD student is a third year. She's Janelle Koala. She has done all of the new imaging techniques for my lab, whether it is the electron microscopy, the super resolution imaging, and the FLIM imaging. Veronica Wu was a master's student who then became our research technician and pitched in every part of this project. And we had an army of undergraduate students who were counting and quantifying for us along the way. Lily Chow is my clinical partner, and we work together on, she helps me try to understand the clinical consequences of these things that we're reporting, and was my co-corresponding author on our diabetes diabetes care paper. Carolyn Mark helps us with our electron imaging, and then Mark Lewis at BioQual for providing those pancreas, and then all of our collaborators along the way. Thank you very much. And this work was funded by the American Diabetes Association and the Harvey Family Foundation.
All right. Thank you very much. It was super cool. We have some time for some questions. Terry Satterfield just said, excellent and important work, Dr. Georgia. Thank you. So I will happy to take questions if anybody has them. I have one, but I want to give other people a chance first. So I'm gonna. Oh, do you have one, Ron?
Yeah, great talk, Senta. I was curious about the primate tissues and localization of the virus in those tissues. Did you try RNA-ish at all, or maybe I missed it?
Oh yes, we did, and it's just the picture just isn't here. But we also used the RNA scope probes against not the spike protein, but I think it's the N protein, or we mix the two of them together, both of those RNA-scope probes, and you can see that the RNA-scope is also present in the beta cells in just our affected islets.
My question is, what do you think the source of the mitochondrial dysfunction is? Is it? Do you think it's primarily from from a viral infection itself, like the virus is doing something, or is it like downstream of that and it's just a phenotype of the infection?
I think that the virus is co-opting the secretory machinery to make more virus, and that's putting a lot of pressure on the mitochondria. That's what I think it is. Then that pressure on the mitochondria is causing that stress and eventual loss of mitochondria. We can, I didn't show this piece of data, but we have like COX-4 staining to show that there's a clear decrease in the number of mitochondria that are present. You can't see them individually, but we call it mitochondrial load is decreased in comparison to our other animals. And so that's another thing that we want to try to be able to do, is to actually quantify that. And so this army of undergraduates has been trying to quantify mitochondrial size and the distance between the cristae to see if we can actually quantify that difference and not just talk about it qualitatively.
Have you looked at like the fission and fusion dynamics and stuff like that with, does the reticulum change?
So we can't tell the dynamics because all we have is fixed tissue. But what we do want to do is that we have permission to receive the tissue now in our BSL-3 facility at USC. And so we will receive the fresh tissue, and we have now begun isolating islets from monkey tissue, which was a process to learn in and of itself. It's not necessarily the same as isolating islets from mouse tissue. And so now that we can get these live islets, we can dissociate them and do those types of studies where we can try to look at mitochondrial fission and fusion. We can actually measure mitochondrial load in and of itself, and do the seahorse experiments to understand mitochondrial metabolism.
Anybody else have a?
Senta, hi, I had a question about some of those, like those virus-related vacuoles. What else do you think they mean about the physiology? Do you think they're related at all to any of the secretion phenotypes too? Or is that, I don't know if there's some way those are tied together?
Oh, I definitely think they're tied together. So the secretory machinery of a beta cell is completely devoted to making insulin. And we see the degranulation of these cells. And so we don't think it's because they're hyper-secreting insulin, but we think that enough of the secretory machinery is being compromised by the virus to decrease insulin secretion.
So,
and then couple that with the shift towards away from oxidative metabolism to more glycolysis, which beta cells just really don't like. We think that is inducing the dysfunction that other people have shown in their in vitro models.
Far out. I mean, the glycolytic metabolism is going to be an ATP, kind of a drain in terms of just not making as much, right? Per unit sugar. But the other question I had was, like, do you think it's a general feature of viral infection of beta cells to cause diabetes then? Because there's nothing sort of special about SARS-CoV-2 here. These are all general viral processes that you've mentioned, right? So that makes me wonder, is like getting a beta cell infection, are there other cells that other viruses that tend to affect them? Is this a general thing? Is this?
So for years, people have tried to track down if type 1 diabetes is caused by a virus. And it's still not entirely clear that it is or it isn't. But the one virus, there's a few, but the most well-characterized virus that can infect your beta cells is an enterovirus. Okay. And that enterovirus does co-opt the machinery and induce acute beta cell loss. So You can see the apoptosis and you can see the cells shriveling up, but it is a different phenotype than SARS-CoV-2. And I'll tell you why I think this is. I think that SARS-CoV-2, like, it really ruins your lungs and like destroys that tissue, but it's not the most virulent. Virulent is not the right word. It's not the most destructive virus that you've ever seen, right?
Okay.
It's not lytic in everything that it infects.
It's not like Ebola where you're going to bleed out of your eyes and stuff.
Yeah, it infects multiple tissues and just irritates the hell out of those tissues. Like it kind of just like scratches at you, and it's an itch that they can't scratch. And when a cell can't scratch an itch, it starts acting up and eventually is going to die, right? So, for instance, we know that people who have SARS-CoV-2, a subset of them, not all of them, lose their ability to smell. Is that temporary? Like, we don't quite know, but clearly SARS-CoV is doing something to those neurons, whether it's injuring them, whether they lose them and they have to be replaced, right? So we think that the same sort of thing is happening to beta cells. They are being injured by this infection. The thing that we don't know is, do they recover from that injury? Or is this going to be a long-standing problem that is going to impact a significant number of people? And I think that this injury can be subacute, right? So if you're the type of person who has plenty of beta cells, you're not insulin resistant, like
Not yet.
Right? But you can get this infection and have this injury to your beta cells and be completely normal. But let you go through a divorce, sit on the couch, eat a bunch of chips for a year while you're trying to drown yourself in soap operas, and then you look up and you might have severe diabetes all of a sudden. And we're never going to be able to say it's like, because you had COVID. But what we are going to see is that this shift in the number of people getting diabetes in the future. And that number could start to be overwhelming for our healthcare system.
Yeah, and I guess it could be for twofold reasons, because of COVID infection and just because of pandemic lockdown, right?
Yeah.
Like just people being, like you said, well, I guess I'll just binge watch another show. I don't want to go outside. You don't want to go out there. I'll just order some more takeout and just watch this. That'll be good.
Yeah, Tiger King, Volume 3, right?
Yeah. Got to find out what happens next.
Got to find out what happens next.
Did Carol Baskin really kill her husband? Jeez.
Okay, well, we are definitely over time now. So thank you again for giving this talk. It was I thought this was just
It was awesome, Senta.
really timely sort of subject, and I know there's a lot of debate, and hopefully it provided me for some clarity. So thank you again.
Yeah, it was just a good talk. It was really nice and enjoyable. Thank you.
Yeah.
Thanks for having me. Have a great day.